So Angela is a Peter O'Donnell postdoctoral fellow at the University of Texas at Austin. And as I mentioned, she was one of the 4 finalists selected from a very competitive pool. So we're very excited about your presentation. Thank you for preparing it in such hectic times and for joining us today, Angela.
All righty. So first of all, I'd like to say thank you again for selecting our abstract for this for these special talks. So for the next 15 minutes, we're going to have a brief tour of our work, and then I'm happy to answer any specific questions afterwards. Jumping right in, the overarching goal of our work really has 2 parts. So the first part is to build a mathematical model. Oh, I heard someone.
Share your screen?
I believe I'm sharing my screen. Hang on. So technical difficulties taken care of there. Apologies for that, everyone. So as we jump right in, luckily it was the first slide. The overarching goal of our work really has 2 parts. The first part is to build a mathematical model that utilizes patient-specific imaging data to predict response to neoadjuvant therapy. Well, neoadjuvant therapy is really just any therapy that occurs prior to surgery. For breast cancer patients, commonly they will undergo 1 or 2 different types of chemotherapy regimens before undergoing surgery. And at surgery, they will determine whether or not the tumor burden has been eliminated, and if there's any residual disease, it'll be removed. So hopefully, if we can build this model that can predict response to therapy, we would like to be able to use it to derive optimal strategies for individual patients for breast cancer. The idea being that we can select a regimen that will give the best overall response for these patients. So what's this imaging data that I'm referring to? Well, it's actually clinical data acquired in the community setting, which is an important note because we tailor our approach such that it may be generally applied in the standard of care eventually. The MRI data that we work with is diffusion weighted and dynamic contrast enhanced magnetic resonance imaging. And this MRI data gives us quite a bit of information about these tumors. So for example, we can identify the region of interest, which tells us how big the tumor is, where is its placement in the breast. We can derive quantitative values for the vasculature, and this tells us how perfuse the tissue is, and we can approximate where chemotherapies might go. And we can also calculate out the number of tumor cells throughout the tumors. So what this MRI data in general does gives us a 3D picture of how this tumor is, how big it is, where is the vasculature, where is therapy going, where are the highest cellular densities, and we want to be able to import all of this into the mathematical model for individual patients. So our established mathematical model is a reaction diffusion model, where n here is the number of tumor cells changing in time and space. The middle term is just logistic growth, where theta is our carrying capacity per voxel. A voxel is a 3D pixel. Since we know the size of the voxels and we have estimates of tumor cell size and packing density for breast cancer, we can fix this value. And then we also have a proliferation map. So instead of having a global proliferation value for the entire tumor, we actually assign proliferation values for each voxel, which gives us a bit of heterogeneity in our simulations, which is nice because we are currently working with only 1 cell type at this time. What's another unique thing about this model is our diffusion term, which is mechanically coupled to the tissue properties of the breast. So here is a cross-section of an MRI, where the affected breast has the tumor highlighted in color. Can you see my mouse? I'm curious. Yes? Can people see the mouse? I just need a nod or a thumbs up, or maybe they can't see it. So in the affected breast, you can see that, yes? Okay. Thank you. So in the affected breast, you can see that the tumor is highlighted in color. And then we have bright gray and lighter or darker gray areas throughout the tissue, which represent fibroglandular and adipose tissues or fat. And these different tissues have different stiffnesses. So so the idea is that when the tumor grows, it's going to push against these tissues, causing different stresses, and which would affect the evolution of the tumor. So we've tried to include this in our model. There's a bit of information here, but you can always go to the paper. But to summarize, basically what happened is by including this mechanical coupling to modify the diffusion, the mathematical model does a better job at capturing basically the shape of these tumors as they evolve in its predictions. The newest term of the model is really this treatment term, and we've since expanded and changed this term to begin differentiating between chemotherapies. And that's because breast cancer patients are commonly given 2 chemotherapies at the same time. So here you can see 2 different chemotherapy terms. where the alpha values are efficacy parameters that we calibrate per patient. We have the C0 drug 1 and C0 drug 2, which are initial concentrations of the drugs that we derive from the DCE MRI data for each dose that they receive. We also have these nice exponential decay terms, which we're able to bound based on literature values for terminal elimination of these drugs. So this is actually pretty simplistic, the way that we're implementing this, but as all modelers know, even a simple change needs to be evaluated to see if you made the model worse or if you've improved its performance. So in our group, how do we go about evaluating our models? Well, we say that we try to evaluate its predictive ability. And the way that we collect the MRI scans allows us to do just that. So here you can see this is a therapy regimen. We have scan 1 occurring prior to any therapy. Scan 2 occurs after just one cycle of therapy, and that's important that these occur early on in neoadjuvant therapy. And then the last scan happens after the completion of the neoadjuvant therapy regimen. So the idea is calibrate the model using very early data in the therapy, and then try to predict response to the therapy regimen at the completion of that therapy. So the idea is here that we can take our 3D simulations from the model and directly compare to the patient's actual response according to their MRI scans. Now, there are lots of different ways we could compare the model simulation to the MRI data, but one way that we do this is we use 3 summary measures of these tumors. So we have total cellularity, total volume, and longest axis. The longest axis is actually very commonly used in the clinical setting to determine whether or not a tumor has responded to a therapy regimen. So here I'm showing you the corresponding unity plots for a cohort of 18 patients. We're on the y-axis, we have the predicted values from the mathematical model. And on the x-axis, we have the measured values according to each patient's MRI scan. You'll notice from the concordance and Pearson correlation coefficients that the mathematical model's predictions are actually well correlated with the actual response that happened within this cohort. And I can tell you that this is an improvement upon previous model forms. Some of you have probably already noticed, though, that we have different colored dots within this graph, in these graphs. We have blue and red. So these are actually 2 different groups within this cohort that we notice. The blue patients are the ones that receive chemotherapies only, and the red patients also received chemotherapy, but additionally, they were given targeted therapies due to their positive receptor status that made them eligible for that additional therapy. And you'll notice with these concordance correlation coefficients that the Pearson correlation coefficients that the mathematical model does not do as good of a job for these patients that also receive targeted therapy. So this suggests that there needs to be an expansion of the model. That being said, before we move on to changing the model, we asked ourselves, is there anything else we can do beyond evaluating predictive ability? Is there some kind of study that we can do that will tell us about the potential of the system? So we said, well, what about generating in silico therapy regimens? So what do I mean by that? So before we were thinking about this as, okay, we're going to calibrate and we're going to try and predict and see what the model says. But what if we change this to just simulate? What if we change the therapy regimen here and see if we can get a better or worse response compared to the standard of care regimen? So we wanted to do this in a logical fashion so that we could compare back to the standard regimen. So we said, okay, let's take the same total dose between scan 2 and scan 3, but change the dosage and timing of the regimen. So here, let's pretend that this regimen is a whole dose every 2 weeks. Well, we could try a half dose every week or a half dose with twice the frequency. Similar, you can think about this as, okay, well, we'll give a third dose with 3 times the frequency. So this way we have the same total dose, but we're changing the dosage and frequency within the regimen. And this is actually plausible to try. And the reason for that is because of the new invention of these take-home infusion pumps that chemotherapy patients can use, where these pumps give a very low amount of chemotherapy over extended period of time to try to mitigate side effects. So we said, okay, let's see what happens. I'm just going to show you the plots for total cellularity as to not overwhelm everyone with a bunch of figures. This first figure, you can see lots of colors. Along the x-axis, we have each of those chemo-only patients that the model did a good job predicting response. Along the y-axis, we have the predicted percent change in total cellularity from scan 2 to scan 3, where a positive value would indicate the model predicts that the tumor would regrow, and a negative value would indicate that the tumor would shrink. So basically, the dots that are the lowest would be the best response for these individual regimens that we're trying. Blue being the standard regimen, and the other colors are these alternative regimens. And you can see within here that if you look at any given column, that there isn't really one color that dominates. So we decided to compare directly. We said, okay, if we compare each of the alternative regimens to the standard regimen, what do we get? And we've created this heat map of what is the additional percent reduction possibly achievable had the alternative regimen been implemented over the standard of care. So the easiest one to digest here is for the fifth patient, this fifth column. And you can see that it's all dark blue. And what this indicates is that none of the alternative regimens were able to outperform the standard of care according to the model. However, if you look throughout this heat map, you can see there are a lot of other different colors, and that for some patients, over 10, 20, even over 40% additional percent reduction was predicted possible for these alternative regimens. Now, this is all very preliminary, but what's really cool about this is we're actually capturing a phenomena that happens in the real world. Certainly, we see the standard regimen does really well for a few of these patients, and that's what we see in real life. Standard of care, you will see patients that achieve a pathological complete response, or PCR, by surgery. However, there are many, many other patients that do not. And what the model is showing that is perhaps if we can build a model based on each individual's tumor and physiology, that perhaps we can select a regimen that will then get us better results, get us a better tumor control on an individual patient basis. So this is kind of exciting work that we're moving toward. However, remember that we had trouble with those targeted therapies, and the mathematical model was not as good at predicting the response for those patients. So if we want to include targeted therapies, we actually need some other information. So targeted therapies and chemotherapies are very different. And one of the simplest comparisons is the size of these therapies. So chemotherapies are very small molecules, whereas targeted therapies are very large. They're actually antibodies. So we need different information about the distribution of the targeted therapies to be able to incorporate this into the model. So we've been working with our collaborators at City of Hope out in California, where they're doing a very unique study where they are collecting positron emission tomography data or PET data as well at the same time for MRI, at the same time as the MRI for breast cancer patients that are also undergoing neoadjuvant therapy. So what is this data that I'm talking about? What does this look like? So here we have two patients, one for each row. We still have our contrast enhanced MRI. We still have our diffusion weighted MRI. So all of the information I told you about at the beginning of the talk is still available. And now we have this copper 64 PET data that tells us about the distribution of these targeted therapies. So this is really exciting. And I'd like to note that while these are just two patients, this work that we were doing was really a test of the entire pipeline that we have. The interlacing of these two different modalities was really a challenge and really exciting and really has not been done yet for clinical breast cancer data. So I just wanted to point that out. So both of these patients received the same therapy regimen, so the same chemotherapies and also the same targeted therapies, which were specifically anti-HER2. So these patients had their breast cancers were positive for the HER2 receptor. And like I alluded to earlier, although they were given the same standard of care, they achieved opposite responses by surgery. So one patient was a PCR and the other one was a non-PCR. Okay, so how do we incorporate this targeted therapy into the mathematical model? Well, the two therapies that these patients actually received are called trastuzumab and pertuzumab. And the mechanism of action for these drugs is actually the antibody will go and bind to that HER2 receptor, and it's thought to decrease the proliferation of the tumor cells. Now, I'd like to note at this time that this group that I'm in, the Yankelov lab, we work and challenge ourselves both experimentally and mathematically at the in vitro and preclinical scales to understand how not just targeted therapies, but chemotherapies in general work so that we can leverage that information into our clinical and tissue scale models. And it's just one of the ways that one of the strategies we use to attack the multi-scale problem that is cancer. Now, since there isn't very much time, and I know I'm already probably going over, I will just kind of overview what we did to the proliferation here. So we still have our proliferation map, like I mentioned earlier, but now we have a function that depends on the concentration of those antibodies that we derive from that PET data. So this will modulate the proliferation, so to specifically decrease the proliferation. And this is based off of our previous work at the in vitro scale. Okay, so how do we go about evaluating the model this time? So actually, we are going to make a comparison between the two different models. We're going to run the MRI-based model. I hear someone. Is someone trying to interrupt? No? Okay, so we have the MRI.
Oh, sorry.
What was that?
That was a 14-minute warning.
Okay, I'm sorry. All right, I'll keep going. So the MRI-based model, we have where it has no targeted therapy, and then we have the PET MRI-based model where we have included this targeted therapy component. We're going to calibrate the model still, but we don't have a third scan to compare to with this data set, so we're just going to see how the two models compare. So the MRI-based model, you can see that it predicts for, if we look at cellularity and volume, it predicts that both tumors will actually regrow from scan 2 to the time of surgery. In fact, it says patient 1 is going to do worse. It's going to regrow a lot. So based on these predictions, we'd say that patient 1 is not going to do well with this therapy regimen. However, if we look at the new model, the PET MRI model, we have that it predicts that patient 1's tumor will shrink, while as patient 2's tumor will regrow, but not quite as aggressively. So not only do we have an opposite prediction here that's easily distinguishable, shrinking versus growing, we also have it saying that no, patient 2 will be the one that does worse on this therapy regimen. In reality, patient 1 was the pathological complete response and patient 2 was not. So this is only 2 patients, but we see this as preliminary evidence that perhaps this modification of the model moving forward, we can, as City of Hope has already been collecting the data, and we're looking forward to see if this trend holds up. And it's exciting alone just to interlace these different modalities of data. So I'm going to summarize moving forward. This is the last slide. The mathematical model, we've seen that it can be predictive of tumor response using the earliest times of neoadjuvant therapy data, and that's important because if we're able to predict whether or not a patient will respond to therapy early on, then perhaps we can intervene early on to change their therapy regimen. Also, that may mean that we might be able to select the best regimen for patients using a mathematical model. We need to continue to gather and process data for larger cohorts of patients to see if those statistics hold up and continue to evolve the model. In particular, we're working on modeling the spatiotemporal evolution of the vasculature to do a better job capturing where the drug therapies will be going. And again, we're going to be leveraging some of our preclinical work for that. And the very last slide here, we got to say thank you to all the wonderful people that work on these projects, all the wonderful funding, and most importantly, thank you to all of the women who volunteer their time and data for these studies. And I can take questions now.